Welcome everyone to the second lecture on metallobiomolecules. Molecules associated with metal ions play a major role in regulating biological process. Depending upon the nature of the other molecular part, it is classified into two categories: proteins and non-proteins. The proteins are further classified into enzymes and transport and storage proteins. Metalloproteins account for nearly half of all proteins present in nature. Metal binding sites are responsible for catalyzing important biological processes such as photosynthesis, respiration, water oxidation, molecular oxygen reduction, and nitrogen fixation. Metalloproteins play many different roles in cells such as storage and transport of proteins, enzymes, and signal transduction proteins. The metal ions in metalloproteins are typically coordinated to nitrogen, oxygen, or sulfur centers that belong to amino acid residues of the polypeptide chain incorporated into the protein. Here you can see the list of metallobiomolecules present in living systems. Metallobiomolecules are classified into proteins and non-proteins. Proteins are further divided into transport and storage proteins and also enzymes. The transport and storage proteins are further classified into oxygen binding proteins, metal management proteins, and electron carrier proteins. The enzymes are further classified as hydrolases, oxidoreductases, isomerases, and synthases. And we are going to discuss some of the very important metallobiomolecules present in living systems, and they are highlighted here. Now, let's see what are enzymes. We know that enzymes are metallobiomolecules with molecular part protein which are biological catalysts in function. Depending upon their functions, enzymes are classified into hydrolases, oxidoreductases, and isomerases. The hydrolase enzyme includes carboxypeptidase and carbonic anhydrase. These are zinc-containing enzymes. The oxidoreductase enzymes include superoxide dismutase, which contains copper, zinc, iron, and manganese. The tyrosinase enzyme, which contains copper metal, and the nitrogenase enzyme, which contains iron, molybdenum, and vanadium metal. The isomerase and synthase enzymes are usually the coenzymes present in body. For example, we have vitamin B12 that contains cobalt metal in it. Now, moving on to the non-proteins present in living systems, the metallobiomolecules with molecular part a non-protein is the non-protein metallobiomolecules. For example, chlorophyll. So, these are mainly transport and storage proteins. The metallobiomolecules with molecular part as proteins have metal ion transport and storage function. They are mainly hemoglobin, which has iron in it. Myoglobin is the storage protein present in humans which contain iron in it. The hemocyanin is the oxygen carrying protein in aquatic animals or other lower animal systems. Cytochromes and blue copper proteins are the electron transfer proteins present in living systems. Now, we are moving on to the zinc containing enzymes, which are very important in bioinorganic chemistry. First, we will discuss the carboxypeptidase enzyme. It is the first metalloprotease and the second zinc enzyme that is being identified. There are actually two kinds of carboxypeptidases. These enzymes that are not metalloenzymes, for example, yeast carboxypeptidase C, and the other one is enzymes that are metalloenzymes. The example is carboxypeptidase A, that is which we are going to discuss in the coming lectures. Now, let's see what is carboxypeptidase A. Actually, it is an exopeptidase. What is exopeptidase? It means that it will cleave only the terminal peptide bond, and it hydrolyzes a peptide bond at the carboxy terminal of the peptide chain. The presence of aromatic substituents at C terminus will favor the hydrolysis, and carboxypeptidase B catalyzes the hydrolysis of basic amino acids, particularly basic amino acids like lysine, arginine, ornithine from the C terminal position of the polypeptides. Now, I already mentioned that the hydrolysis of carboxypeptidase A occurs only at the C terminal residue that has an S configuration, and also at the end, which is carboxyl group free. At neutral pH, in absence of the enzyme, the above reaction occurs at a very low rate constant rate. However, when the reaction is catalyzed in presence of carboxypeptidase, the rate constant can attain value of 10 raised to 4 per second. So, here you can see the mechanism of the hydrolysis by carboxypeptidase A. Here you can see that it hydrolyzes the peptide linkage here, and here the carboxylic end is free and it occurs the hydrolysis at this portion. Now, we are going to see the structure of the enzyme carboxypeptidase A in detail. I already mentioned that it is a zinc containing enzyme. It consists of a single protein chain of molecular mass about 35 kilodaltons. It has zinc in +2 oxidation state in the active site. You can see here it is Zn 2+. It is ellipsoidal with a cleft on one side that contains the zinc ion. Zinc coordination number is 5 with imidazole nitrogens from 2 histidine residues, histidine 69 and histidine 196, and we have 2 carboxylate oxygens of glutamine. These are the 2 carboxylate oxygens and a water molecule. Here you can see the water molecule. So, this is the structure of carboxypeptidase enzyme. Now, let us see the geometry of the enzyme. It is having distorted a pentacoordination geometry. Here you can see it is pentacoordinated. Many acidic and basic amino acids like asparagine 144, arginine, tyrosine, arginine 127 and glutamate 270 are also present in the active site and is very important for the activity of the enzyme. The active site cavity has a hydrophobic pocket that accommodates the non-polar R of the C-terminal amino acid of the substrate undergoing hydrolysis. This shows the mechanism of hydrolysis of the polypeptide by the carboxypeptidase enzyme. Mechanism of the carboxypeptide enzyme hydrolysis. The bound water is converted into a nucleophilic hydroxide which then attacks the carbonyl carbon of the sessile peptide bond. The catalytic mechanism involves a nucleophilic attack by the hydroxide on the carbonyl carbon of the C-terminal peptide bond on the carbonyl carbon of the C-terminal peptide bond. This is a nucleophilic addition reaction. The first step is the positioning of the substrate on the enzyme. The enzyme interacts with the substrate via hydrogen and van der Waals interaction if the terminal group is aromatic. If the terminal group is aromatic, the enzyme will interact with the substrate via hydrogen and van der Waals attractive forces. The interaction of arginine 127 with the carbonyl of the terminal peptide bond increases the Lewis acidity of and make it more susceptible to nucleophilic addition. Then the water molecule bound to zinc performs a nucleophilic attack on the carbonyl carbon of the C-terminal peptide bond with simultaneous transfer of a proton to glutamate 270. At this point, the glutamate 72 bound to the zinc ion becomes monodentate from bidentate, maintaining the 5 coordination of the zinc ion. This process is called the carboxylate shift. Very, very important. And this process is called the carboxylate shift. The protonated glutamate 270 now transfers the proton to the amide group of the terminal peptide bond. This causes the collapse of the tetrahedral intermediate, resulting in the formation of a free carboxyl terminal amino acid and the shortened peptide chain with its terminal carboxylate group bound to the zinc. A water molecule will then add to the zinc ion. Finally, the cleaved peptide leaves the zinc ion and gets back to the original penta coordination. Now, let's move on to the next zinc-containing enzyme, that is the carbonic anhydrase. It is the first zinc metallic enzyme identified. It catalyzes the reversible hydration of carbon dioxide to form bicarbonate ion. It catalyzes the reversible hydration of carbon dioxide to form bicarbonate ion. They play a very essential role in biological processes like respiration, photosynthesis, and help in maintaining pH. The carbonic anhydrase enzyme occur in animals, plants, and several bacteria. It is a single chain polypeptide with molar mass about 30 kilodaltons. Now, we are going to see the structure of this enzyme. The active site of the enzyme is located at the bottom of a cleft in the enzyme. The enzyme is composed of about 259 amino acids. It is containing a dipositive zinc ion. A dipositive zinc ion activates the water molecule prior to the reaction with carbon dioxide. This zinc is coordinated to the imidazole nitrogen atoms of 3 histidine residues. You can see 3 histidine residues to which the zinc 2 plus is being coordinated. The 3 histidine NH protons are involved in hydrogen bonding. The 4th coordination site of zinc is occupied by the water molecule. The 4th coordination site is occupied by the water molecule. Now, let's see the geometry of this enzyme. It is having a distorted tetrahedral geometry. The active site pocket also contains ordered network of water molecules and other amino acids like threonine 199, glutamine 106, histidine 64 that are essential for hydrogen bonding, proton transfer, and for binding the carbon dioxide substrate. In this slide, you can see the mechanism of carbonic anhydrase. Actually, the reaction occurs in 4 steps. The first step is the deprotonation step, followed by carboxylation. Then the formation of a ring structure will occur, and finally the addition of water molecule occurs. Here you can see first the deprotonation occurs, then you have the carboxylation occurring, then there is the formation of the ring structure here, and finally the addition of water molecule will occur. Actually, the Isidine 2+ is a Lewis acid. The zinc ion present in this enzyme is more acidic than the zinc present in carboxypeptidase A. Histidine being neutral and less basic than glutamate ion, it makes zinc more acidic in this enzyme. We have already seen the mechanism of carbonic anhydrase. It involves 4 steps mainly: deprotonation, carboxylation, formation of the ring structure, and finally the last step is the addition of water molecule that will result in the formation of the bicarbonate. The binding of water to zinc reduces the pKa of water from its normal 14 to 7, and this leads to the deprotonation at neutral pH to form the strong hydroxide nucleophile. Now we are moving on to the summary of this module. Metallobiomolecules play many different roles in cells, such as they are part of storage and transport of proteins, they are part of enzymes and signal transduction in living systems. The metal binding sites are responsible for catalyzing important biological processes like photosynthesis, respiration, water oxidation, molecular oxygen reduction, and nitrogen fixation. We have also gone through the structure, function, and mode of action of zinc-containing enzymes, particularly carboxypeptidase A and carbonic anhydrase. Hope you are all benefited from the lecture. Thank you.